was from Cambridge in neuroscience. And after travelling around the world, I came back to Australia and was fortunate enough to meet a range of different neuroscientists who I was very lucky to and honoured to work with, and Gary is one of those. That was in about, correct me if I'm wrong, Gary, but I think that was around 1995-96 that I first got to work with Gary and his team. And at that time, you were working on research projects at the Austin, doing NMRIs, PETs, positron emission tomography, which is outdated almost now, isn't it? I don't even know if we use it ever more. I don't practice in the field of neuroimaging, which is, you will find through Gary's talk, how we now look at the brain, what all those colours mean, how we do different tests while they're in those big horrible tubes, the big white tubes. Has anyone had an MRI? Or yeah, it's pretty terrifying stuff, isn't it? No, it's fascinating. You loved it. You enjoyed it. That might be why you're in that tunnel. No. Now to introduce, I will now get formal. Sorry about that. But nevertheless, also over that time, I came to know more of Gary's particular approach to neuroscience, and I would say, given also my international experience, that he is an international expert in what's known as neuroinformatics. That is like the information from the brain, neuro being the brain, informatics being informational data. And he'll be modest and say, what are you talking about? And he'll probably scold me when he comes up to the microphone to say, don't be ridiculous, I'm not an international expert. But I can say with heart, hand on heart, that of all the people I've met across the world, he is an international expert, and we're very honoured to have him in Melbourne. And it's just as well he got married and had kids and had to stay here, because we would have lost him to better salaries overseas. There's no doubt. Now, Gary is currently the Principal Research Fellow and Professor and Director of the Monash Biomedical Imaging Research Platform, currently being established at Monash University, surprisingly with that name. Before that, he was across the road on a corner office, almost overlooking us. He's only just revealed to me, I knew I said I was going to get serious, but his story's in between. He just told me that he's moved from university house to graduate house in 2008 to have lunch, so yay. However, he's now moved to Monash, so the convenience of walking across the road to have lunch has decreased somewhat. And that was evident in the phone call I got urgently at, 6:05 saying, I'm stuck in traffic. He was previously an NHMRC Principal Research Fellow at the Howard Florey Institute, which is now known as the Florey Institute of Neurosciences.
Nearly Florey Neuroscience Institute.
Yeah. There we have it. And I was very fortunate to sort of work in the same building when I was with Neuroscience Victoria and Neurosciences Australia, and again we sort of touched bases. I don't think we did research together, but we certainly did sort of integration of neurosciences projects, and one of the platforms that Gary was responsible was coordinating all these different types of imaging the brain across Victoria. Did you want to actually give a talk, or shall I just keep going? And you He was the 2008 Churchill Fellow. He's been working in this field for over 25 years. He looks young, but he's really old. Yes, and he's also been incredibly good at looking after the livelihoods of researchers. So he's got $1 million worth of grants. He's kept academics in employment, in particular research academics, and he's published in the best journals in this area across the world. And these are not easy journals to get published in. We're talking, you can come up right up the front, and I'll really make you feel really awkward. So tonight, Gary will explain the technicoloured images. I wanted to call it the technicoloured image interpretation of the brain or something like that, but visualising the brain is the title. Please turn off your mobile phones. I forgot to say that. So he'll be explaining what they mean and how our scientists, but more importantly our clinicians, neurologists, psychiatrists, psychologists and other clinical people interpret these images to get you better, or to diagnose you, or to progress and monitor your condition after you've had, for example, a stroke, or you might have multiple sclerosis, or you might have Parkinson's disease or a psychiatric disorder. So please put your hands together and welcome Professor Gary Egan.
Well, good evening, everybody, and thank you, Gary, for that overly kind introduction. Yeah. Most of what she said was completely untrue, of course. But I have known Gary for a long time, and I think it was 1995 or thereabouts. And in fact, I first met Gary. I think, well, the thing that sticks in my mind is... I want to take my jacket off. It's really warm in here, so I'll just take my jacket off. I was invited by Gary to present at a symposium in Churchill. And for those of you who know your Eastern Victorian geography, of course, that's down in the Gippsland area, beautiful spot. And I had no idea that there was a Monash University outpost at Gippsland. And so I and there was a weekend involved, so we had accommodation. I thought that'd be lovely, go down to Gippsland and spend a weekend. I think it was in sort of late spring, really lovely. So I went down there, and here is Gary and her colleagues doing phenomenally exciting stuff with movements, how people planned movements, and how you differentiated brain areas that were involved in either grasping with the hand or grasping with the mouth. And we did an imaging study together, and we published that. I didn't actually look it up this afternoon, but I was saying earlier that I tried to find the slides of that particular research project so I could share it with Gary here tonight. But unfortunately, it was in the days before computers, and so they literally are little, Ektachrome type slides. So there we go. I couldn't show it tonight. Just shows you that everything pre-sort of internet and computers doesn't exist anymore, which is, well, not quite true, but a bit sad. So look, thank you very much, Gary, for the very kind invitation to speak here this evening. And I would like to say at the outset that I'm very happy to take questions and comments throughout my presentation. I'm a little bit overawed actually, because this is the first time I've ever done this, and by that I mean I've never actually had people pay to go to listen to one of my talks before. so I thought, wow, I have to really take this seriously tonight. And so I've gone through, it's a little bit of a sort of a walkthrough around research that I've been involved in over the last 15, 20 years, and also trying to sort of look at where we came from 100 years ago and where we might be in the next, well, 5, 10 years. I certainly wouldn't think beyond that sort of time frame. So I'm going to sort of give you a little bit of a walkthrough over some of the last century of research that is focused on how we image the brain, how we visualize the brain, and how we understand the brain. And of course, this is the brain that we're going to be talking about. It's mostly the human brain. In fact, I think every slide I'll show will be around the human brain, but of course neuroscience works across all of the different species, and many of the great discoveries have been made in very simple organisms which have as few as a few thousand neurons, and nothing as complicated as the human brain. But of course the elegance now with the modern scanners that we have, MRI scanners and so on, means that we can actually image people's thoughts, and even we can image, and I'll mention this towards the end, up to 8 seconds before you think of the thought, we can actually see it happening in your brain, which is really quite amazing. But it's been shown scientifically that we can now actually predict where there will be, perhaps not a thought, but perhaps a motor action and that sort of thing. So we're going to be talking about the human brain, and Australian scientists have been very prominent in neuroscience over the last century, and particularly if we go back to 1963, I think it was, or 1962, when Sir John Eccles, a great Australian neuroscientist, was awarded the Nobel Prize in Medicine for his research on figuring out the neurochemical transmission of brain signals. And before that, people knew there was an electrical component, and then it was John Eccles' work that identified the synapse, the little gap between successive synapses, or between brain cells, where the chemicals are transmitted. And so this slide's particularly appropriate because, you know, Australian scientists have been, you know, thinking about, imagining and dreaming of the brain in the Australian context for over a century. And in fact, one of the great neuroanatomists of the past century was an Australian, and we'll come on to him shortly. But I wanted to kick off by saying, well, actually, let me just go back a tad. So I wanted to start by showing you, I'm going to be talking about, you know, our living brains and how we look inside people's minds almost now, not quite their souls, but inside their thought processes. And of course, that's one level of description, which is what we call systems approach. But there's another level, which is right down at the cells. And this next video I'm going to show you is a video that sort of highlights that all the things we're talking about at the sort of brain level really operate right down at the cell level, at the individual neurons and individual cells. So this video is from the Allen Brain Institute in Seattle, which was founded by Paul Allen, one of the co-founders of Microsoft. And he's funded a $100 million research centre about 8 or 10 years ago to look down into the details of individual brain sections. And these incredible images that have been constructed here are from taking sections of post-mortem human brains and then going in and imaging them at incredible resolution at 1,000th of a millimetre or at micron resolution. And then seeing individual neurons, like you can see here, seeing the complexity of how different brain cells connect to their adjacent or very distant brain cells. And then to look at the dynamic or the sort of plasticity of how these different brain cells can connect with each other, how they signal to each other. And you can see these sort of flashes of light, which I grant you are simulations. This wasn't a living brain when these images were taken. But it sort of highlights to you that what we're looking at in brain imaging, brain function, is a truly amazingly complex system, absolutely complex. Probably, as most people sort of use the phrase now, you know, the most complex system that we know, more complex certainly than the universe, because the principles or laws of physics that describe the universe are greatly simplified down to very simplistic laws, or very complex but a few laws. Whereas in the brain we know that the complexity of this incredibly interconnected and massively parallel connections between all of these different brain cells is what enables us as humans to have the enormous breadth of capacities and of functions. And so when we look at brain images, of course we're looking at, this is a human brain, and this is a section which is a coronal section in this orientation, and we see here, perhaps I'll just take it back and stop it right there. We see all of the grey matter around the edge, and we see the deep white matter, which is where the axons are that connect the brain cells that are distributed mostly through the grey matter, or in these deeper grey matter structures, the central midbrain and other internal structures where the connections can pass through. And so it is, yes?
You did invite a question, and in the movie there are other cells that were moving quickly across the screen in between and through the medium. And would you just like to quickly comment on them as well as, because you've explained the neurons? Yes,
so there's other cells that support the neurons. There's astrocytes and glia, so the cells that provide both myelination for the axons which connect the different neurons together. And then there are capillaries where the blood is flowing through the oxygenated hemoglobin, and that was kind of what you saw trafficking along were blood cells right down at the single capillary level. So the arteries going to arterioles and then perfuse right down to the individual capillaries, which is sort of one hemoglobin red blood cell molecule wide, and they traffic across and give their oxygen off into the tissue, the brain tissue, and it's transported into the neuron where the metabolism occurs as a result of the brain activity. So yeah, thank you for the question. So let me go on and sort of just again, I guess, highlight that I'm going to be talking up here at this level, which is the systems neuroscience or systems approach, and recognising that we go right down to individual molecules that communicate between an axon terminal here and then the next part of the next brain cell, and this synaptic cleft or the synapse is again equally important in terms of describing or visualising how the brain works. But the challenge of that area is that it's at a millionth of a millimetre. These things are happening very quickly, the level of a millionth of a second or less. And so that is still beyond our reach to be able to image, and certainly from outside or without sort of invasive techniques. Whereas in the last 20 years, as you'll see as I go along, the ability to look at the whole human brain at about a millimetre resolution and at about a second resolution temporally is what we've been able to achieve, which has been really quite remarkable, and also, you know, very exciting to have been able to work in this space during that time. So, where did we start? What you saw from that Allen Brain Atlas or Brain Institute video was work in the last 5 years, which has really taken a sort of almost converted neuroscience, and Kerry might disagree with me on this. I think it's taken neuroscience from being what I'd call a cottage industry into being a really mainstream research activity with industrialised approaches. The Institute there has employed, you know, hundreds of people from day one. They've done, you know, thousands of brain sections, and they've really made it a real challenge for scientists in university contexts and even large research institutes like the Florey Neuroscience Institute to be competitive. It's like competing with Microsoft or Apple for that matter. They're really doing it in a very big way. But of course, it hasn't been like that for very long, and we're adapting as scientists to that environment. But when it started, it was in fact a very individual and not even a cottage industry, but, you know, a sole worker type of industry. And the person who's, I guess, attributed or recognised as having done the first images of the brain in some way that were usable by the rest of the community was Corbinian Brodmann, who did work in Germany in the early part of the 20th century and published these maps, which are called the Brodmann maps or Brodmann atlases, back in about 1915, I think it was. Correct me if I'm wrong there, Kerry, 1920, something like that. So nearly,
people I'd like to add to that.
There's quite a few. And the person I mentioned earlier, the great Australian neuroanatomist Campbell, was one of the key other contributors to this work. And what these people did was took, you know, post-mortem brains, because there was no way of looking inside and visualising what the brain was. You could just see the outside. But to try and figure out that it's not just a like a sort of a soup or a gel, there is really internal parts of the brain and individual parts. They looked at the histology or the microscopic detail, and this is an example here of looking at a section of the brain as you go through the grey matter on the outer layer. And they were able to show different layering and different cell densities. For example, here in this area of the brain, the motor cortex, which is the bit that controls how you perform motor tasks, they were able to identify in layer 5 these giant BET cells, which were the output cells that go down the corticospinal tract. And so Brodmann and his colleagues were then able to say there's a part of the brain, which is this bit, which is where all the motor control happens. And they didn't quite know it was motor control then. What they knew was that it was a special part of the brain that was different than the others. And it wasn't until they could see in patients who have lesions or people that had tumours that they could then figure out indirectly that those people had, you know, disrupted motor function, and that was the part of the brain that had been damaged. And so you could work out indirectly that this sort of area controlled motor, and then this region here in grey, which is numbered 1, 2, 3, is the sensory area. So all the tactile input or somatosensory activity or brain function is received into the brain in this area. So this was the first visualisation, which is probably a bit of an overstatement, but it's the first way that we could see that the brain had special parts to it, and it wasn't just some sort of uniform, squidgy tissue. And then, since then, many other areas, and there's still a lot of debate between neuroscientists in some areas as to whether there's sort of one or two or perhaps three areas. How different are the cell densities in different layers as you go across different areas of the brain. And you know, that's always good grist for the mill for putting in for grant applications and getting funding to continue on with the research. But of course it is really crucial because the human brain is so enormously complex that there is highly specialised subdivisions of these different anatomical areas. And of particular relevance is the frontal lobe here, which you can see is divided into about 7 or 8 regions, but in fact is incredibly more complicated and diverse than that. So this was the first sort of visualization of a brain atlas, and then this is a kind of a montage I put together to sort of jump forward almost 100 years and see all the other types of brain images that we can now collect. And I'll come back to the different images on this slide as I go through my talk. But you can see up here now we can actually image people doing motor tasks, and we can see in different brain sections whereabouts there is increased brain activity as a result of doing that motor task. And down here you can see we can now plot from this rotating brain with diffusion MR imaging these tracks which identify that white matter region that connects one grey matter area to another grey matter area. And so this is sort of the jump that we've been able to make in the last 100 years or so. Okay, so visualising the brain, where it began. Well, it really began with this first observation by William Röntgen over 100 years ago, 1896, when he did this photographic plate exposure of his wife's, it's his wife I believe, with her wedding ring, of her hand on a plate by virtue of the fact that he realised that radioactive materials, which had just been recently discovered, emanated something which then could cast an image like this on a photographic plate. And of course that was an absolute breakthrough discovery, and he received the Nobel Prize for that in 1901, the discovery and use of X-rays. And he put those X-rays to good use generally, but of course they were unfortunately misused in a number of cases, and there were problems associated with that. But it was the first observation that there was a way of looking inside the body when the body was still alive, and that was a phenomenal step forward. A long time later, and most of you would probably remember when the first computer tomography scanners or CAT scanners became available, and these scanners now, which are very widespread and incredibly fast, were really a technological development by putting computers together with X-rays, because a CT scanner literally just uses X-rays, but it collects them in a ring, and it then enables images which are not just projection images, but in fact cross-sectional images through the object. And that was the big step forward that occurred in the early 70s. And Geoffrey Hounsfield, who's a British physicist, was the person who did this by combining computers with X-rays and invented the CT or CAT scanner. And he got the Nobel Prize for this work in 1979. And some of you may remember that incredibly significant step forward. So we sort of were starting to get through the 20th century at this stage with computers and X-rays. And then even more recently was another step forward, which is Kerry mentioned, with magnetic resonance imaging. We've now come into sort of the modern day in a sense, where we can take images of people without using X-rays. We can just look at the magnetic properties of tissue. And these images are able to look not just at structure, because the X-rays really get attenuated by bone, but we can look at function. And I'll show you some of that as we go along. So here's a nice small MRI scanner. So those people who've had an MRI scanner, what I can say to you is that it's actually getting more fun in an MRI scanner, because the MRI scanners are getting smaller, more compact. Some people are shaking their head, but They are getting more compact and friendlier. In fact, the one that we have at Monash University that I'll show you at the end of my talk is a nice blue colour. It's got a little light in it that even sort of wafts off and on. So they're getting very, very comforting, actually. And I was at the Monash Medical Centre this afternoon at the MRI facility there. And they're really, they're terrifically well configured now to be patient-friendly. But we use the MRI scanners for research, and we collect brain images like this one that you can see. This is now a sagittal image, so I'm taking slices running this way through the brain. And you can see, if I sort of stop it somewhere, you can see the orbits over here, the eyes. You can see this part of the brain is the cerebellum. And down the back here is the visual processing area. And the bit that I was pointing out earlier, the motor cortex would sort of be up here, where the output areas are. And this image tells us still about structure. So in fact, we've come a long way, but not actually that far, because it's still just how the brain is structurally made. It's not telling us what functional areas are mapped into these brain parts. And the thing that I remember someone mentioning when functional MRI came out was that you could actually use functional MRI to tell if your volunteer was still alive or not. It's probably not a good thing to advertise with the volunteers that we ask for our studies. But whereas when you do structural imaging, you actually can't tell if this is a live person or not. So MRI has come into this type of structural imaging, and the big step forward compared to CT was that this image actually can resolve this grey matter area that I showed you earlier on, and these other deeper grey matter structures here, from all of this white matter, which is the parts that connect the brain. I'll let that run through, and that's an image from our scanner at Monash, which we turned on 2 months ago, which was very exciting for us. And here's another image from that scanner, which is sort of the same. This is now a coronal image, so it's running sort of front ways. And you can see as we go through that, I'll just jump back and show you that one again. It's a bit scary. I know who this is. It wasn't me. I'm not that scary. But the interesting about this one is that MRI actually has lots of different magnetic properties that it can detect. So in this image, all of the water in the ventricles, the cerebrospinal fluid, is very bright, and the brain tissue is kind of dark, or the white matter is dark. Whereas in the one I just showed you earlier, it's kind of the reverse. And this is because there are many different magnetic properties that exist in the brain and in biological tissues. And so that's one of the new features with MRI compared to the CT or CAT scan, as I mentioned a little bit earlier. So these discoveries have been recognised with Nobel Prizes, as I mentioned. Röntgen's Prize back in 1901, which was absolutely outstanding. And then in 1979, Jeffrey Hounsfield and Alan McCormick's Nobel Prize in Medicine. And then in the last 10 years or so, Paul Lauterbach and Peter Mansfield from the UK for essentially using magnets and lots of fancy electronics and computers to collect these images of people without no radioactivity, no X-rays, no invasive interference, so completely safe, and now obviously used very widely. Okay, so I've told you all about structure. Here's an example of, I guess, the key point I was making earlier. Homer had some sort of structural brain image there. He had a very big functional brain, and he had very big representations of parts of the brain. That's what we would call a very large representation, and a very big beer representation. And so that's what a lot of researchers now do in brain research with imaging, is try and figure out where these functions sit on top of this anatomy or structure that I've been talking about. So I'm going to just, I hope I'm not going too scientifically here, and I'm going to go slightly more and then I'm going to back off. I just wanted to give you a sense of one experiment we did about 10 years ago that kind of really pushed the envelope here. One of the works that Kerry mentioned, where we were able to get published in a leading American journal. And it was sort of coming back to my earlier point. We tried to figure out in individual people where their functional area was, sitting on top of this structural area, Brodmann area 4. And the way we did it was we asked people to lie in an MRI scanner for about 6 hours. So that was really going beyond the pale. Yes.
You haven't mentioned the cases where they ask you to stop breathing, and the cases where they don't. And could you explain to us why?
Sure. Well, if you're getting an MR where they're asking you to stop, to inhale, or even a CT scan, a single inhalation, it's a chest or thoracic scan that they'll be doing.
No,
for the brain as well. Okay.
Yeah,
So so there's also pulsatile motion in the sort of brainstem area, which is because of the cardiac cycle. You can't do anything about that. You can gate the scans, you can collect the ECG signal and then correct a little bit, but not a lot. But you can ask people to at least not move their head up and down as they're respiring, and that's the main reason.
Yeah, except that you can't, because if they fix your head completely,
yeah,
you're just going to move up and down.
That's true as well, but some people are pretty determined to move when they inspire. And in fact, yeah, they're not actually literally screwing you in, so you can move. And the resolution we're scanning now is about 1/4 of a millimetre resolution, so people can certainly move that much, or maybe a millimetre. So, yeah, so we asked people to lie still. I don't think you volunteered for this study, did you, Kerry?
I've never.
You've never volunteered?
Yeah, never.
Well, that's handy. Good to know. I've got a good study for you. So we... Good, I'm glad to hear it. So we repeatedly scanned them many, many times, and that meant that we could actually get images that were significantly better spatial resolution 10 years ago, before the scanners could really do it. And at the same time, we asked people, and here's an example of, it's not a real subject, this one, but we asked people to move their finger, and you'll see this right index finger was a motor task. So they have to activate this brain area and make this part of the finger move, and then we studied whereabouts that was in this anatomy. So the point I'm trying to get here is that, like, we know from Brodmann's atlas 100 years ago that there is this very specific area, but that was actually done in one post-mortem brain. And what we don't know is across our population, the variability, where exactly that brain area is structurally, and then even more so, when people do brain function, is it always onto the same structural brain area, or does it vary? Is it sort of out on the outer edge for some people, deeper for other people? And on an individual basis, where does the function in our brains actually map to? So this study was to figure that out in 6 different people, and you can see in this one, when the motor task was running, we got this area of activity, and when we asked them to do something different, which was to sense a grating, so they had some rough paper under their fingers, we then saw, I don't know if you just saw that, but there was an area here, which is where you sense things coming in through your sensory, somatosensory input, and it's not in the same area, it's in the post-central gyrus, this is the pre-central, and this is the main sulcus that divides the frontal lobe from the parietal area of the brain. So we're able to see where motor was, where sensory activity is, and now we're sort of doing it almost microscopically in living people, and that was so exciting for us. Because we were asking people to do this for so long, we couldn't do it in a large number, so we really started treating our subjects like our laboratory monkeys. But I was one of the subjects, and of course as researchers we all volunteer for this work. It's about figuring out basic neuroscience, and we all chip in and lend our brains to science. And the thing that we particularly did as well was looked with the anatomy. You can see up in this area of the brain there are some sort of lines running through, and this was a signal of the different cells in different layers of the grey matter, and that was how we could see structurally, a la Brodmann's work, whereabouts the structure and the function were correlated or co-located. So that was one area of research that we did with the Children's Hospital scanner, actually, up at the Royal Children's Hospital about 10 years ago. It was terrific good fun. And then a little bit more recently, we sort of said, well, we can see what I would now call sort of static function. So when you ask someone to do something all the time, same thing, you can see where that bit of the brain is, but it's just like a constant level of activity. And of course, when you ask people to do that, they do it for about 2 seconds, and then they get bored. And then they start doing something else, like not the task that you've asked them to do. They think about what their shopping list is, what they're going to do when they get out of the scanner, and all sorts of other things. And so we realised that, of course, our simplistic approach of asking them to where motor function is or not was really simple. And it's a much more complex system. And as I was saying at the outset, the dynamic nature of the brain was what became more and more apparent. So we designed this experiment where we could ask people to do a task and then look at how they did that continuously. And when I show you this now, you'll see this is a little plot of when they're being asked to do the task, and then a brain area that sort of spikes on and then off, and then sort of comes up and spikes on again at the end of doing the task. And so it's not just an off-on thing, it's not just a switch. And when we look at this set of sections, you'll see that there's different types of responses as per the colours. And so here's a flush of brain activity right at the beginning when they start the task, so you use a whole lot of brain areas. And then now they're in this point where they're sort of in a constant, just doing it and probably thinking about the shopping list or what they're going to do when they get out of the scanner. And these other brain areas become deactivated. And the brain areas here in the thalamus and the cerebellum. And here's this motor output area that I was talking about earlier. They are sort of in a steady state activated. And then right at the end of the task, when now you have to think about, you know, oh, I've got to stop doing the task. Now suddenly there's a whole flush of activity right through the brain again. So this was the 4th dimension. So now we're actually doing 4-dimensional functional imaging. Not just static function and not just structure, but time-dependent functional activity. And this is sort of really, I guess for me at least, where it's kind of at the moment. There are further advances. I suppose the limitation of what I've shown you is that in spite of this sort of video showing that, it's only being sampled every 2 seconds. We're not able to sample it at sort of a 1,000th of a second, which is really how the brain works, or even at tens of milliseconds.
What restricts your sampling time?
Well, it's really the technology at this stage. It's the MRI scanner. And we collect these images of sort of the whole brain in 2 seconds. And it's the brain activity dependent images, or the blood flow dependent images. Look, I'm very hopeful it will, and one of the interesting things here is that...
Gary, can I ask you to repeat the question?
Oh, sorry. Sure. So the question was, would it be likely to become faster over the next few years? And the interesting thing is that it's now... MRI was a sort of specialist medical technology in a sense, and now there are people coming in from many electrical engineering disciplines, and the group here at Melbourne are very strong in radar, which is actually a similar approach mathematically, but for remote sensing, looking at things, you know, like a long way away and tracking them. And MRI is an example of near-field sensing. And so they're now actually adapting their radar techniques towards near-field sensing with MR, and can do it very, very fast and cleverly with algorithms. So we can actually scan, in fact, faster than that, but there's still technical development to push it beyond about a second or so. Yeah.
In the example you see up there, if you ask the patient to just do mentally the exercise without physically doing it, what happens?
Well, now it's very interesting you asked that question. I'm wondering if that's a Dorothy Dixon. So the question was, if you just ask people to imagine doing the motor task, will they still activate all these brain areas? And as I mentioned at the beginning, with Gary, we did an experiment which was, you know, which brain areas are activated when you grasp something with your fingers, or which areas are activated when you grasp something with your mouth. And I really wished I'd have found this slide of this experiment, because the way that we tested this was with a little bit of inducement. So we used those red gelatin snakes and we asked people to grasp those with their mouth or with their fingers, but we also asked them to imagine grasping them with their fingers. So we had 3 conditions in that experiment. And it turns out that in fact you do, for an imagined grasp, you do activate the areas which are sort of back here in the parietal lobe, which are planning areas for making a motor task. And you can even activate some of the sort of areas associated with perhaps pre-motor areas, but you don't activate the motor output area, this area 4 that I pointed out. So you set up a system in the brain, but you sort of don't press go, and therefore you don't activate motor problem area 4. Okay, so, whoops, go on from that. Okay, so you probably got a little bit of a flavour that this is a field of lots of different sciences that are all kind of coming under the umbrella of neuroscience. And my background actually is in physics. So when I met Kerry and we started thinking of experiments, you know, it was like lots of different ideas flowing around. Kerry, as she said, has a neuroscience, a hardcore neuroscience background, looking at cells and so on. But we're now, you know, and I've learned enough neuroanatomy so I can sort of find my way through the bits of the brain. But it's a very exciting area, and this interdisciplinary nature, the complexity of the brain, the fact that we've got technologies to measure, we've got people with biological understanding from the neurobiology side, people with information processing from computers about, you know, how information can be transmitted, etc. And that's really why this area is kind of today absolutely in the avant-garde of sciences. Okay, so I'm going to move on and go to some other types of brain structure and function, and point out to you that the areas so far I've discussed have been the grey matter, where the brain cells are. But I haven't told you about how they're connected to each other. And again, at the early video you saw it's enormously complex. And it was only about 10 years ago with MRI and a particular technique, diffusion MRI, where we could start to look at how water actually is differentially trapped into these major white matter tracts in the brain, and enables us with MRI to see the tracks of the axons between extended distant brain cells or neurons. And so this image is a sort of classical image that's been published, which is of the inner parts of the brain. We're not talking about neurons at all. We're talking about the deep white matter tracts. And these are connected between the 2 hemispheres through this region here, which is the corpus callosum, or the part that connects the 2 brain lobes. And in each lobe there's also tracts running down the spine, corticospinal and other neuronal output. So this whole area now is called the connectome, and that's because brain imaging researchers didn't have an ohm. So we had to invent one that was kind of roughly relevant to what we were doing. So there was genomics and proteomics and metabolomics, and now we've got connectomics, and no one else can do that. Only brain imaging people can do that. So we're very proud of that. I didn't invent the word, I wish I had. So it's a massively exciting new area. And I'll give you a little bit of a feel for what this means. So here's another view of the brain, a medial sagittal section, as we say. So this is now looking at the inner surface of the right hemisphere. And this is the corpus callosum, which is the white matter tracts that connect the 2 hemispheres together. And here's the spine, and here's the pons, and here's the cerebellum. And pretty much everything you can see in the cortex is the outer fold, which is the grey matter layer. And you have to cut into that to see the underlying white matter. And it's only in the corpus callosum, where it is the white matter through the hemispheres, where you can clearly see the axons. And when we use MRI now, we can come up with these sorts of maps, which I sort of showed you a moment ago. But the tracks are really phenomenally beautiful, and this is where I think Kerry was inspiring me to talk about the Technicolor brain, because this is sort of the avant-garde of MRI imaging, where these images show the different directionality of the fibres in the brain. And the colours encode direction, that's why they're there. Whereas in this image we only have intensity. In this image we have intensity and direction. And so we code the direction with the colour. And the colours here are, blue basically represents fibres that are running up and down, or sort of superior, inferior, dorso, sort of ventral. And then green represents, in this case, fibres that are running sort of posterior anterior. And in another view I'll show you the red colours represent fibres that are going between the hemispheres, so in and out of the board. So the colour in these images really has some meaning. And I'm going to step through this a little bit, probably quickly, just to highlight, which I haven't actually said, but thank you, which is that MRI measures magnetic properties, but it really looks at the properties of protons, and the protons are primarily, thank you, primarily in the form of water, because that's the most abundant molecule in the body. We're basically mostly water. A few other elements thrown in and mixed up. And so MRI senses protons, and it senses water, and it senses the cellular environment of those water molecules. And this little plot here, which I'll kind of run through again, shows you what happens with Brownian diffusion of water, but in the context of constrained, sort of cylindrically shaped tissue or axons deep in the white matter. And when we do diffusion imaging, we then create models, and this is like a ellipsoid shape that describes how water is preferentially diffusing in one direction. And we use that to say, well, we think underneath that there are these thin axons or fibres or white matter fibres. And when we have that information, we can then produce different maps, and in this case, in each voxel, we get the directionality plotted, as you can see with these little red arrows. And so now what we're finding is in each voxel in the brain we can determine what is the optimum direction of water diffusion, which tells us which direction the fibres are running in the brain. And that's an amazing sort of step forward, because now we can see incredibly detailed orientation information in living human brains. And as I mentioned before, we can then colour code these directions and see, for example, here, left, right in red, these are the corticospinal tracts here in the external capsule that are blue, and then these major lines running from the front to the back are in green. And this is the recent exciting technique, which I showed you at the start, how these images of brain tracts can be constructed. There's the Technicolor version, and then here's one particular pathway that connects the cerebellum up through the thalamus into the somatosensory area. So this work, of course, is great fun to do, exciting and challenging, but it's also got a clinical benefit, and I wanted to share with you one application, which is in a project with Huntington's disease patients, where we've studied the fact that there are brain areas that degenerate early on in Huntington's and are in fact involved in motor function. So we know of Huntington's chorea and motor dysfunction. And so by all of this research I've sort of been talking about can be applied to understanding our diseases, and particularly neurodegenerative diseases, where we can look at the evolution of brain changes over time. And in fact, what we've done is looked in a deep brain structure, which is this area here, which is called the caudate, and how it connects into these cortical areas can be deduced. So here's another view of this internal structure that we're interested in, the caudate, and then a part adjacent, a bit more lateral, which is the putamen. And one of my PhD students, India Bohanna, who was an incredibly talented young woman and now working up in Cairns, she was able to figure out how to generate tracks from these cortical areas shown here in blue, or orange or red as the motor, pink as this temporal area, and how they connect into either this deep structure, the caudate, or this adjacent structure, the putamen. And she did that and came up with these maps, which were then, in 10 different people, a probabilistic atlas of whereabouts, for example, the temporal areas connect into this fronto or rostral area of the putamen here, or actually no, it's the caudate, and not really represented at all in the putamen. Now this is a bit technical, but the point I want to make is that these tracked images have enabled us to see these structures deep in the brains of Huntington's patients, and we've been able to see where there's particular cells that are dying as a part of the Huntington's disease, the medium spiny neurons, and identified very specific subparts of these structures by doing this tractography research. And obviously the application here is to look at patients well before they have clinical symptoms, and in fact as early as now about 18 or 20 years, we can see in these very specific small areas, we can see that there's been these neurons that have lost or died. And unfortunately we can't do a lot about it at this point, but it highlights where the kind of origin of the disease process is, or the pathogenesis. And the next step is to trial therapies in people who are gene positive, who are known that they will develop Huntington's, but not for another 20 years or thereabouts. And so we can give them therapies well before any major symptoms arise, and therapies which we can then monitor with this sort of imaging and see if it prevents the loss or atrophy or dysfunction of these brain areas. And so that's a key area of work I was doing here at Parkville until a year or so ago, but which is continuing on in the community here, which we call clinical neuroscience, where all of this technology is applied to understand the disease process and then to monitor the use of new therapies, experimental medical therapies in these patients. One other area of work which I've had a lot of involvement in with Trevor Kilpatrick here at Melbourne University was in using MRI and diffusion MRI to understand changes that occur in the optic nerves here in people when they present with loss of visual acuity, which could be a sort of a marker of the very earliest onset of multiple sclerosis. So people who get lesions in their central nervous system sometimes have those lesions firstly, or at least present to a doctor when those lesions are in the optic nerve, because they get a loss of visual acuity. They might have had lesions elsewhere, but there was no obvious symptom, and they wouldn't present to a GP or a neurologist. But we know that people with loss of visual acuity may go on to have multiple sclerosis, and therefore it represents an opportunity to look at people or patients at the very first point of onset, and then to study at that point what's happening in the brain of people who may develop MS. So some of the work I've described to you was to use diffusion imaging in individuals to map the white matter tracks that come from the back of the orbits as a result of looking at a visual checkerboard, and look at how this signal or this information is transmitted along the axons from the ganglion cells on the back of the retina. And in normal subjects we know that those axons are heavily myelinated, so they have a good insulation sheath, whereas in people with optic neuritis they can have loss of the myelin around those axons, and therefore reduced intensity of signal and sometimes crosstalk between adjacent axons, and so they have loss of visual acuity. So that's a second example of how this sort of sophisticated research can be used in clinical neuroscience. So I'm coming to the last part of my talk. I don't know if there are any questions or comments about that.
Well, I'd like to ask a question. Has there been any effective mapping done that you've done in other areas?
In disease states?
Or just in normal people, both?
Yeah, so as I mentioned earlier, the visual areas have been sort of, in fact, sort of visual neuroscientists would argue, so the question was, has there been sort of mapping research done in other brain areas beyond the motor and the smarter sensory?
We're trying to just the emotional areas, if you like, of the brain, if they've been
Affective,
right,
as in
yeah.
emotional psychology.
Right, yeah. So that's an excellent question, and it sort of highlights that what I've been talking about, what we call primary cortical areas, so areas are involved in the senses essentially, so vision, touch, motor.
No, I was thinking more of DiMaggio's work on emotions.
Yes, I take your point. And there's, so the areas in the emotional brain, so the insula, the deeper structures, have been mapped with sort of more interoceptive phenomena. So the point of difference here is that when we're looking at primary brain areas like auditory areas, visual areas, these are what we would call extra or external receptor areas. So they're getting signals from the external environment But there's a whole other set of brain areas more centrally which map the internal feelings that we have, both emotional but also what you might call instinctual, which relate to one's sense of comfort in the body, which might be because you're nicely sated or you've had a drink, or perhaps discomfort if you haven't had a drink, or haven't been able to satisfy other urges. And we've been looking at some of these areas actually here at the Florey. I didn't mention earlier, but the Florey, the Howard Florey Institute, as it used to be known, was founded by Derek Denton, and he actually is a physiologist, and so his whole research program over the first 40 years of the Florey was basically about body fluid balance and salt balance research. And it turns out that that's one of the areas that's very strongly mapped in these insular areas, because thirst is one of these instinctual behaviours that, or thirst drives instinctual behaviours. And so we were the first group with Professor Denton to map brain areas in living humans that responded to increased demand for thirst. I won't tell you how we did that. It's not particularly comfortable. There were volunteers, and it was done again up at the Royal Children's Hospital. So that's one way of saying that there are sort of two levels of the emotional brain. The sort of instinctual parts, which are interoceptive driven processes, and then there's this link between the emotional, perhaps more social emotional areas, which come in through olfactory regions, for example, inferior frontal areas, and so on. And it's a very active area of research, and Damasio's work has clearly led the field in understanding that there's a whole other part of the brain. In fact, emotion is a bona fide area of scientific research. Before his work, which was in the late 90s, certainly, probably, I was a bit of a skeptic myself that emotion wasn't a bona fide area of research scientifically, but it's very well mainstream now. And the other point to make there perhaps is that not just social emotions, well, yeah, they're still social emotions, but again, what I would call instinctual emotions, such as fear, have been directly mapped into structures, the amygdala and different nuclei of the amygdala, and their connections back into these central areas in the insular and cingulate, which are those deep grey matter areas, is again still an active area. It's still being worked out, actually. A bit of a long-winded response, but I'll press on. So that's what we've been doing with visualising the brain, and now I'm going to share with you what we're going to be able to do very soon. And when I say very soon, I mean in the next 18 months, because here at Parkville there's a next generation of MRI scanners being installed in the Melbourne Brain Centre. A 7 Tesla MRI scanner for looking at human brain research. And so the point about it is that when we go from the original MRI scanners, 1.5 Tesla, now they're conventionally 3 Tesla, every time we double this magnetic field strength, it's like when the astronomers or astrophysicists look into the sky and increase the baseline of their telescopes. Every time they do that, they can look further into space, further back in time, with greater resolution, because their triangulation base has increased. And in brain imaging, what we do is increase the magnetic field strength that more sensitively maps the magnetic properties of brain tissue. And it's like looking further back into the detailed history of time. So here's an image of the next generation of MRI scanners. I should perhaps stop there. And so this is now the back of the head. It's the occipital lobe where vision is processed. Here's the top of the cerebellum. So you perhaps remember the image I showed you earlier of the grey and white matter. Well, this is now twice or 3 times as high a resolution and signal. And now we can actually see some of these layers I talked about earlier, but this whole image was collected in 6 minutes. So whereas we took 6 hours to do it 10 years ago, now we can do it in 6 minutes. Or at least this scanner, which is in Korea, where I did some work a couple of years ago, was about the 3rd scanner in the world, I think, at that time, and was able to plot out, you can see vessels. Some of these images have signal loss because there's deoxygenated hemoglobin running through vessels which come through these grey matter areas. And this is the main sulcus through the occipital lobe, which is the calcarine sulcus, And we know that this is where the areas from the orbits from the retina map directly into this brain tissue here. So that's very exciting. We can also, that will move forward, perhaps the next one, here we go. We can also look at some other brain structures. What I was telling you before about mapping from the outside to the inside, here's some now higher resolution. It's almost like having a microscope in a living brain. These are living people and 6 minutes. So we're now going to the next level of possibilities for looking in the brain. Here's an example between the brainstem and you can see the cerebellum, so like it's amazingly better. This is just anatomy or structure, and lots of other images which I could go through at another time. So another area of future research, I mentioned the Allen Brain Atlas. They've done mouse brains, human brains, spinal cords, non-human brains, and it's a really phenomenal large data set. Kerry mentioned what we call neuroinformatics or neuroscience informatics, which is essentially like the Facebook of neuroscience, basically. So all of the researchers put their data on the internet, and the Allen has been one of the major contributors, and then other people access that data, images, or the particular thing that the Allen people do, which is unique in their case, is that they do gene expression mapping. And so they correlate different genes that are expressed in different sections of the brain, and here's some different genes expressed throughout cortical areas in these couple of post-mortem human brains. You can't do gene expression mapping yet in vivo. You can take a blood sample, but you can't map it to the different anatomical parts of the brain. So that's probably beyond the limits of any technological developments. But it highlights sort of the next phase. What we call imaging genetics, bringing imaging that I've shown you together with all of the functional genomics that is an equally, if not more, active area of research. So just to wind up, two more things I wanted to share with you. Basically where my new home is, where I live these days. This is quite a way away, 20 kilometres or so, and right next to the Australian synchrotron. So I have good company. This is the Monash Biomedical Imaging Facility, and we have an MRI scanner doing the sort of research I've explained to you. Also some other scanners which are radioisotope molecular imaging techniques. And the particular thing that was done at the Australian synchrotron, which is shown here as a sketch, is to build a medical imaging beamline. And there's 25 synchrotrons around the world, and ours is one of the newest, but in fact we're the only one with an external beamline which comes out in such a way that you can do medical imaging research using a synchrotron. It was decided by the Australian National Health and Medical Research Council, with the strength of biomedical research in Australia, and particularly Melbourne, that they invested 20 million into this external beamline, and that's nearly completed. In fact, here's a top view. This is a Google map view, I think. You can see the synchrotron, so it's quite large. It generates X-rays, and so sort of back where we were 100 years ago, but we're now not just doing X-rays from sort of radioactivity or radioactive materials. What we're doing is having an electron beam that shoots around and then is made to emit X-rays of a very specific energy, single energy, and then collimated down to a few microns or 1,000th of a millimetre resolution, and then can be targeted onto structures like individual cells in tumours, in cancers, for example. So the research at this facility will focus on radiation therapy protocols and research. It comes into this building, which is now linked into the Monash Biomedical Imaging building, where we have the MRI scanners, and in here we do X-ray imaging. It will be with clinical research and even patients at some point, probably in the next 3 or 4 years, but right now it's focused on preclinical or animal-based research. So here's some of the scanners that we've installed at MDI. Here's this nice blue MRI, if anyone would like to try it, very welcome. It's nice and wide actually, it's a bit wider, and with modern trends we've had to get a bed that can take up to 250 kilograms. So that's a feature of this particular scanner. And these are the other scanners that we have which are for the animal research work. So a couple of images. This is basically a mouse scanner. It does molecular imaging and CT imaging, and you get images of the kidneys of rats, for example, or up there some other CT images. And so here's an example of some brain or head images. I didn't really show you any CT images of the brain because you can't see anything in them. All you can see is the skull, that's where the bone is. I'm glad there's no CT radiologists here, they'll probably take exception at that. But these are the sort of images now of a mouse, and you can see the exquisite detail that this sort of little scanner I just showed you can get. It's about 20 microns, so it's about a 50th of a millimetre resolution, which is pretty good and probably adequate for a mouse. But what I showed you at the synchrotron is another level again. The synchrotron is at 1 micron, so a 1,000th of a millimetre. And here's an image of a mouse now with synchrotron X-ray imaging, and that's just amazing. Like, that is extraordinary. And so this is really the only place in the world we can do this work at this level. The people in Melbourne have worked in Japan on their synchrotrons for the last 6 or 8 years, and also in Chicago, but as I explained, ours is a dedicated long external beamline, and so this will clearly become the centre of this sort of research globally. So we can look at bony structures with X-rays. You can see the shoulder plate of the mouse, which is quite remarkable. And we can also do another technique which is called phase contrast X-ray. And I won't go through what that means, but it really comes to the fact that we can look at soft tissue in living organisms. So we can actually image the lungs of a rat, and we can fly through. So this is reconstructed. We're going down through the bronchioles, right down ultimately into the small parts of the lung of a rat. And we can see the sort of bifurcations that you saw there, and the sort of rib cage on the outside. And now we're sort of pulling back out of that. So we can, at 1 micron resolution, look inside living animals with extraordinary detail. The work at the synchrotron, actually done in Japan, but by researchers at Monash, has focused on imaging lungs and has been used to improve the treatment of lung disease in premature babies. Because babies in neonatal intensive care units almost invariably have lung disease, and the way that they're ventilated is a critical part of how they can be treated and cared for with lung disease. And the researchers here looking at lung images in animal models that were actually born whilst in the scanner, so had the umbilical cord cut whilst they were actually being scanned, revealed the fact that the ventilation strategies were inadequate, and they've been adjusted on a global scale worldwide over the last couple of years as a result of this research in lung imaging. So it's enormously significant what the work is that's being done there. Okay, my last points. Visualising the brain and imagining the future. Well, it's a bit hard to imagine the future, so I thought I'd just leave you with a couple of ideas that now go into the next creative application of this sort of data and technology, and working now with some artists who are using biomedical imaging, MRI scan data, to sort of conceive of what this means in a broader social context, how the community gets involved in this research, how it interprets it, and how it can be communicated, particularly to the wider public. And this is work that's been done nearly 15 years ago by an Australian artist, Justine Cooper, who was really an avant-garde artist who was in the vanguard of all this research. And I first saw her work really amazingly in the Ballarat Art Gallery. If you've been there, you would have perhaps, you would have noticed this sculpture, which is upstairs, and it's in the middle of a fairly large room, better size of this room. And I was sort of looking at this Perspex block. It looks like this. It's about maybe 3/4 of a metre, sort of cube. And as I was looking at it from afar, I could see, like there's something in there, and I'm not sure what it is, but I kind of went over and had a look. And you can see there are gaps between each one of these Perspex plates. And so then you can look in, and when you do that, what she's done is taken a CT scan of her head, these axial sections, and she's magnified it 4 times, and then placed it on these Perspex blocks, or plates, to make a Perspex block, to make a translucent human head CT image. It was really intriguing and it caught my attention. So then I started to follow her work, and this is what she has done more recently, now using MRI data. You can see this on the web. It's called RAPT, R-A-P-T, and this is now the sort of fusion of art with biomedical imaging science and biomedical imaging data. So I'll just finish there and let you listen to this, about a minute or so of this video. Thank you for your attention.
The nerve glaucoma or the retinal damage, seeing as what, 80–85% of inputs to the brain are visual, is there an effect on the structure and function of the brain? Surely it doesn't remain unchanged if it is not getting the information that it needs.
Well, that's absolutely. It's a terrific question. So did everyone hear the question? Do you want me to repeat the question?
Yes.
So the question is that in people who suffer loss of vision, because so much of the brain is devoted to processing visual input, what happens to those areas of the brain when people lose vision through glaucoma or loss of sight? And this has been a really interesting problem that's really now been solved by functional imaging. People did studies in individuals who were blind from birth, and they could see that those very extensive visual areas in the occipital lobe had been reprogrammed to do other functions. And so you can now see that signals are coming into these posterior occipital areas which are not related to visual input. So areas, for example, with braille reading, when people are reading braille, those somatosensory areas are mapping down into areas that would otherwise be used for visual function.
Do you call that plasticity?
So that's a great example of plasticity, yeah.
But does it happen in aging adults?
Well, are you able to use that bit of brain for some other function?
Yeah.
I
do
think
the other parts which don't get visual input, you know, can they change to...
So there is generalised plasticity of the human brain. So people with stroke have been able to have areas, rehabilitation therapies can regenerate function that might otherwise be lost. Obviously motor and sensory function are some of the areas that have been shown to be remapped. And then there's even great examples of people with large tumours in one hemisphere where in fact, whereas of course mostly your right body side is controlled through your left hemisphere and vice versa. People have a lesion in one area for, say, motor function, in fact have motor for both hemispheres controlled from the other hemisphere. And so that's a classic example of plasticity. But these things can happen, typically that happens in children or when people have tumours as children. As to whether you can use your brain function that is not required for a given primary cortical or primary sensory input, I think the jury's still out on that a little bit. It's a complicated problem because you can't really do controlled experiments. You can in animal models, and animal models suggest that there is much more widespread plasticity than we have really expected or thought could be possible. But in the context of sort of what you do with that brain area now that you're not using it for vision, that's, I think, not really known now. Yeah.
Can you make a comment on what is known about the function of the brain as we go through the process of learning something?
Yeah, good question there as well. So the question is, do we understand which brain areas are being activated perhaps more during learning processes? And that's actually, that motor experiment I showed you with the hand was exactly that. It was a motor learning experiment. I didn't go through the detail. We asked people to do one of two tasks. One task was to do a thumb and finger opposition task, which was one 3, 2, 4, 1, 3, 2, 4, and they learnt one of these tasks and they weren't allowed to learn another task. And when they came back after 4 weeks, we re-scanned them with the task that they'd learnt, which was 1, 3, 2, 4. And we also scanned them with a new task, which was, in fact, 3, 1, 4, 2. And they weren't allowed to learn that one. And so we could clearly see brain areas which had got increased blood flow, so what we would call angiogenesis, or blood, vascular blood, capillary increased blood flow in deep brain structures initially, such as in the thalamus, in the area that controls the sensory input from the tactile stimulus, and also in the motor cortex. Again, in that sort of structure I showed you, there's a very clear representation of where each finger that controls function is mapped, and we could see increased angiogenesis in areas involved in the finger areas, and not just immediately adjacent for wrist or elbow. So it showed that neuroplasticity in these very controlled experiments exists in humans and is evident by looking at increased blood flow, which then often sort of diminished again after the acute learning phase. So by the time, so if you think of a musician who's learning an instrument, I mean, it's difficult, they're having this learning process, and angiogenesis is involved remapping the sort of extent of function. Once they've acquired the skill, then there can be again a reduction in the representation and a reduction back to normal blood flow. So we really see sort of what we call functional and anatomical correlates of neuroplasticity and learning.
One more question, Rosie? I've worked in acquired brain injury and then moved on to spinal cord injury. It makes me the life of the party. But I just wondered if there had been any key learning in either acquired brain injury rehab or for people working in that area or spinal cord injury rehab.
Yeah, so in terms of sort of these techniques I've been talking about tonight, there is another technique which sort of goes the opposite way, which stimulates brain. All the things I've talked about have been measuring brain activity by one measure or another, or brain structure. But technique transcranial magnetic stimulation is where you can put a coil, electrical coil, and induce magnetic fields that will penetrate into the brain, which will then generate electrical fields and currents in the cortex of the brain, the grey matter. And so this is a way of stimulating, and it can be either to interfere with sort of neural activity, which is a sort of a large amplitude, or it can be sub-threshold, where it sort of raises the threshold point of brain function. And the benefit of this whole thing, it comes out of some of the animal work again, where you can use these stimulation techniques to look at plasticity and look at long-term potentiation of brain activity. And it highlights that there are, I guess what it comes down to is that we need to use approaches that mimic the natural learning and natural brain plasticity timeframes. So how people learn, what the frequencies are of these stimulations that need to be given, over what duration of time, and then how frequent over a period of days or weeks. And all of that sort of research around stimulation of brain activity is a very active area at the moment. It's still, I would say, the only area that's been used extensively, two areas. One is in treatment-resistant depression, which has been done at the Alfred Hospital by Professor Paul Fitzgerald. And that's been very successful in people who have got diminished brain activity in their frontal lobes. They've been able to upregulate activity in about 50% of the patients who had treatment-resistant depression, which is terrific. And the other area is in stroke again, where there's an area which may have had what we'd call the secondary part of a stroke, or the penumbra, which has got reduced function and can be stimulated to have increased function after there's been a recovery from stroke. So I think it's pretty promising, but it's complicated, and so it's taking a while to figure out the right stimulation regimes. Yeah. Okay, thank you.
Thank you, everybody. I'd now like to, thank you, Gary, of course. I'd now like to call on David Sparks to give a vote of thanks.
Well, Gary, one thing that comes to mind is that we all, or maybe just the age group, that we're all here tonight, but we all tend to know of the terminology of MRI and CAT scans and things. It's almost becoming a community language, so maybe not the younger generation, the older ones more interested in these things. But it was interesting how you developed your talk and gave us an insight of the history of how this, the basic principles of being able to remotely detect the images of the brain, non-contact, non-invasive techniques and so on, which I guess we're all very pleased with. So no one likes to go to hospitals for those, some with a knife in their hand. But I was particularly impressed with your talk tonight because you mentioned electrical engineers. You went up in my esteem, but it does raise the issue, though, that I guess your work is really very much related to a multidisciplinary team of being able to get to the level of research that you can share with us tonight, because I guess you need the computer specialist who can pull all these signals together and make these wonderful visual images to get the message across as to what's happening in there. You need the engineers and physicists to create this machine which has such infinite fine resolution that gives you the degree of study that you need to pick up what's going on there. And you've given us good examples of that tonight. You had examples of early detecting machines versus the more modern 7 Tesla intensity machines. So I can well imagine that the medical-oriented people are keenly looking for their collaborators to break the frontiers, to allow you researchers to look at things in more detail. The geographical map of the brain was very interesting for us all, I dare say. I think we've probably heard occasions of this in our day-to-day life, where different parts of our brain are located in particular physical locations for their to view the sort of structure and the function of the brain. So I thought that was very interesting analysis you made there. And of course the motor and the sensory concepts that are there as well. So with that, in saying thank you to Gary, I'd like to mention something else if you'd do us a small favour. Because here at Graduate House, our research department has come up with this particular material, which we know we've established is a gentle relaxant in small quantities. But what our researchers are concerned about is the effect when large portions of it are taken by the human body. We are aware that on some people it causes violence and other people it causes sleep. So we're hoping that you might take this away and use it fruitfully in your endeavours.
Thank you very much.